Okay, so hello everybody. My name is Shai Elkayam. I'm a master degree student in the lab of Yaron Orenstein at Ben Gurion University in Israel, and I'm presenting today my research named DeepCRISTL, which is a deep learning model that aims to predict the on-target efficiency of CRISPR gene editing. So before I present what is CRISPR, let's give some background and motivation. So as you all probably know, the human genome is made of 10 to the power of 9 nucleotides, which is the 4 types of building blocks that build our genes. The specific sequence that form each of our genes is what makes us who we are. It chooses the color of our eyes, our height, our gender, and so on. But what if we could manipulate nature and choose ourselves the sequence of our genes? Theoretically, if we know what gene sequence is responsible for breathing underwater in fishes, or what genes grow wings in birds, we could change our genes to give us those abilities. And I believe it will be familiar for some of you. So what is CRISPR? CRISPR is a gene editing tool, technology, which gives us the ability to manipulate almost any gene as we like. Using this technology, we can insert new synthetic genes or delete unwanted gene sequence. This technology has already been used in many applications, as we can see in the graph. For example, it has been used in the food industry to produce vegetables that are immune to some diseases. It has been used in human surgery to treat genetic disease, and in many more. In November 18, a Chinese biophysicist has created the first CRISPR editing in human babies, and by synthetically modifying their genome, he prevented them from being able to receive the HIV virus. And even though his experiment had made waves in the scientific community, a few hours after he presented his work, he has been arrested by authorities, since CRISPR editing in humans is currently restricted in most of the world and need a lot of approval to be done. So I'm sure that we are going to see many more CRISPR applications in the near future, and those applications may sometimes look like science fictions coming true. But before we could see superheroes such as Aquaman or Angel going down the street, there is still many research to be done. So how does the CRISPR mechanism work? To cut the DNA in a specific location, researchers need to create a synthetic sequence of 23 nucleotides called guide RNA. That matches the area of the genome they want to cut. The only restriction to the guide is that the last two nucleotides must be GG, and therefore we are able to edit the genome in almost any location we desire. To the gRNA is connected the enzyme called the Cas9 that is responsible of creating the cut of the DNA. The gRNA with the Cas9 is injected into the cell, and when it locates the exact sequence of the guide RNA, the Cas9 cuts the DNA in that location. And as any process in nature, there is a probability for it to achieve success, and this probability is called the on-target efficiency. This is where my part as a computational biologist comes. The aim of my research is to predict the on-target efficiency of a given gRNA. The input data for our model is the gRNA, which is 23 nucleotides, where the last two of them must be GG, and the labels are the on-target efficiencies. Here we can see the on-target efficiencies of 3 different CRISPR experiments that has been done with the same gRNA, but in 3 different enzymes. There is 2 types of experiments data that we use to train our model. The first type of experiment is called high throughput data. And in this experiment, we can receive a lot of on-target results on different gRNAs in the order of a few tens of thousands. But the on-target efficiencies are less correlative with real CRISPR editing due to technical reasons of the experiment. We have used the 3 experiments of this type that are done on 3 different enzymes, and we refer to them as wild type, ESP, and HF, high throughput datasets. The second type is the endogenous experiment, where this experiment reflects better real CRISPR editing, but is also much more difficult to be done, and as a result, we can get less data points from a single experiment. We have used 11 different data sets from this type, and their size are in the order of a few hundred to a few thousands You can see the table. You can see in the table the data sizes of each experiment we used. One of the things that we notified when we observed a high throughput dataset is that the percent correlation between the three enzymes is relatively high. And therefore, we wanted to combine all three datasets and use them to train the same model. There are some classic machine learning models such as the XGBoost model that aims to predict on the endogenous dataset, but those classical machine learning techniques doesn't leverage the high throughput datasets to better perform on the endogenous dataset prediction. And the currently state-of-the-art model that named DeepHF is using a transfer learning technique to pre-train on the high throughput data and then fine-tune the model to predict on the endogenous datasets. But there are two major drawbacks in this model. First, it trains a pre-trained model with only one of the high throughput datasets instead of all three of them. And the second is that it uses a transfer learning method that is not optimized for this type of data, as we'll see further. We had two main goals in our research. The first was to improve the predictions of the high throughput data over the previous state-of-the-art model named DeepHF. And the second goal was to build a model that is trained on the endogenous dataset, but can benefit the information and the general patterns from a model that was previously trained over the high throughput data. For this task, we tested different transfer learning approaches. In addition to the gRNA sequence, we also compute 11 biological features directly from the sequence that is known to affect the gene editing on-target efficiency. 4 features of melting temperature, a binary feature that indicates whether a stem loop exists, 3 features informations regarding the GC content, and 3 features of the free energy of the secondary structure. For the deep learning model, we have used the bidirectional LSTM network where the biological features are concatenated to the LSTM output and inserted into a fully connected layer. While in the DeepHF model, the authors has trained the model with only one type of enzyme high throughput data, our first improvement to the model was the multitask model that combines all 3 high throughput experiment data sets. And to inform the model from which data each input came from, we used the 3 inputs of one hot encoding. The second improvement is the ensemble model that is comprised from 10 identical models that the only difference between them is that they were trained with different initial weights. And the output of the ensemble model was said to be the average of the 10 models. We refer to the ensemble model that has trained on the high throughput data as DeepCRISTL pre-trained model. We first search for the optimal number of models in our ensemble model. So in the x-axis, the x-axis represents the number of models in our ensemble model, and the y-axis is the Spearman correlation of the prediction and the true label on each high throughput dataset. And we can see that after about 10 models, we get convergence, and finally we choose 10 to be the number of models in our ensemble model. The predictions result was tested with a Spearman metric. We can see here the predictions result on each of the high throughput enzymes datasets and the improvement that was done using the multitask over the single task and by using the ensemble model over the single model. So after we finished optimizing the pre-trained model, we wanted to train a model to predict also on the endogenous dataset. But the problem is that in each of the endogenous data experiments, the amount of data is between a few hundred to a few thousand, which is not enough to train a deep learning model. Therefore, we wanted to use our pre-trained model that already learned some general patterns from the high throughput data and fine-tune it to predict on the endogenous data. So we compared between 4 different types of transfer learning approaches. The first one is the most common approach where we retrain only the last hidden layer and the last layer weights. The picture describes in green the weights that are being fine-tuned and in gray those that are kept fixed. But while this approach is commonly used in computer vision tasks where the pre-trained data has a lot of similarity with the target data, and the model needs only a small fine-tuning to fit to the target data, in CRISPR data, this is not the case. Even though there is some correlation between the high throughput data and the endogenous, it is not enough, and therefore the training only on the last layer is not enough to teach the model to predict on the new data. So we tested another approach where we retrain all the model weights. We refer to this approach as the full transfer learning approach. We also tried another approach where we train all the weights besides the weights of the embedding layer, since this layer only learns how to map the one-hot encoded neurons into another vector representation. The problem with the last two approaches is since in the first few epochs the loss function is quite high, the weights of the model can change dramatically and lose the general patterns they have learned from the high throughput data. Therefore, we have used a different transfer learning approach that we refer as gradual learning. And in this type of transfer learning, we first train the model using the last layer approach. And then, and even though the loss function is high, only the last layer weights change dramatically, but all other weights is kept fixed. And then we create another phase of full transfer learning that let all the weights be fine-tuned. But now, since we already trained the last layer, the loss is already low, and the weights doesn't change dramatically, letting the model keep the general patterns of the high throughput data, but still fine-tune all the model weights with the new endogenous data. We also added two baseline trainings for comparison. The first one is the no transfer learning approach, where we only train the model with the pre-trained data and didn't fine-tune it with the endogenous data. The second baseline is the no pre-trained model, where we train the model from scratch over the new endogenous data. So for training the model, we randomly divided the complete data set into 5 iterations. In each partitions, we used 70% for train, 10 for validation, and 20 for test. In each iteration, we first did a 10 cross-fold validation to search for the optimal number of epochs. And we used the early stopping with 15 epochs to stop the model after it stopped improving. Then we combined the train and validation data and train the model again on each of the 10 pre-training models. So here you can see how the loss is changed during the gradual learning approach. In each of the phases, we can see that in the first phase of the last layer described in red, in the first epoch, the loss is relatively high, but then after only a few epochs, it converges. And the last layer approach doesn't improve the loss very much. The more bright colors is the 15 epochs of the early stopping that we have used in our training. So we can see that after 15 epochs, that the model didn't improve. The last layer approach stops, and then we move to the green section that describes the gradual learning approach. We can see how the loss of the model is kept improving dramatically from the last layer training. So we finally compared all of the transfer learning approaches on different endogenous data sets, and we can see that in all of them, the gradual learning approach has won. We also compared ourselves with all state-of-the-art models. We can see that DeepCRISTL outperformed all of the models in all different endogenous data sets. We have also wanted to check how the amount of data affects the performance of the pre-trained model and the fine-tuned models. In the left, we can see how the pre-trained model is improving by adding more high throughput data points. We can see that after about 40,000 data points, it starts converging. And in the right side, we can see how the fine-tuned model is affected by the amount of high throughput data points that train the pre-trained model. And we can see that even though that the pre-trained model has converged, The fine-tuned model has kept improving after 40,000 data points, suggesting that the model is able to learn some general patterns from the high throughput data and use them for predicting on the endogenous data. So for receiving biological insight on the model, we have used silencing map algorithm that uses stochastic gradient descent to find the input sequence that maximizes the output. We have first initialized the input of the model with a uniform representation of the 4 nucleotides, keeping the representation of sequence being fixed as 1. In each iteration, we receive a prediction from the model and compute the derivative of the prediction with respect to each cell. Then each cell of the matrix is being modified in the direction of the gradient, and we repeat this process until the input matrix converges. Finally, we compute the sequence logo representation from the final matrix using the following formula. And we can see from the logo in each of the positions how each of the 4 nucleotides affect the final on-target efficiency. So we generated a sequence logo representation from each matrix and received the following results. First we search for the sequence that maximizes the pre-trained model, and then we generated a sequence logo for each of the endogenous experiment. We can see that in some positions there is a constant pattern in all experiment, teaching us the importance of this nucleotide in this position, where in other positions the nucleotide is random. So let's see some examples. So for example, G in the 20 position has maximized the model in all endogenous datasets. T in position 14 has maximized most of the models. And these two results is matching previous studies. We also find some new insights, such as C in position 18, which maximizes most of the models. So for future work, we believe that the model can be improved by combining epigenetic markers, such as RRBS and open chromatin. We can also combine all endogenous datasets to train one model over all of them, and this can also benefit the model. And also changing the architecture to transformer may lead to great improvement, as shown in recent studies. And I want to finish this with a sentence that I once heard: "From science fiction to reality separates only time." And I'm convinced that this technology will lead to results that currently seems imaginary. But until Marvel and DC Creature will come to life, there is still work to be done. And we hope to see our model helps and integrates in the development of CRISPR technology. And finally, I want to thank, first of all, to my supervisor, Yaron Orenstein, for his guidance, his academic insight, and his positive feedbacks. I also want to thank the Israel Innovation Authority and the Israel Scientists Foundation for supporting my research. And last, thank you for listening.